My name is Clare Edminster, and I'm a rising senior at Colgate University. And this summer, I worked in the Gore Lab with my mentor, Aaron Farmer, to develop a method to be able to predict phenotypes in maize. So just as a little bit of a background, a phenotype is any observable trait that an organism has, and being able to measure and predict phenotypes is essential for the advancement of agriculture to be able to provide nutritious and healthy plants to a growing global population in the time of climate change. But measuring and predicting these phenotypes is a very time and labor-intensive task. So we developed a pipeline that begins in the field and ends with a phenotypic prediction that increases the rate and efficiency of predicting these phenotypes. So we do this essentially by capturing images of the field, converting those images into numbers, and then converting those numbers into phenotypic predictions. So the two types of images we capture, one captures information spectral information at the canopy level, and one captures architectural information at the sub-canopy level. So we have two different types of information captured by these proximal remote sensors, and both includes valuable but unique information. We then convert those images into numbers using an autoencoder, which is a type of unsupervised deep learning. And the autoencoder essentially reduces the dimensionality of the inputted image and spits out a vector of numbers that represent that information. And we can see here in figure 1 that not only do these numbers, these latent features, contain biologically relevant information, they actually perform better at predicting phenotypes than the currently standardized accepted way of doing so. So our method has promise for applications in the future.
Time for a question? Okay, you did a great job. I'm just wondering, does the canopy or sub-canopy method produce a better prediction than the other?
Yeah, that's a great question. And we actually asked that. We wanted to see, well, we have the canopy and we have the sub-canopy. How can we increase the predictability here? And we actually did so by integrating the information from the two into one large data set of numbers. And actually this figure here shows that using the integrated data provides an even better phenotypic prediction model than using either one alone.
Excellent. Do you have any questions?
Why you use maize as an organism to make this prediction? How about other systems, say model system, Arabidopsis? Because I would say Arabidopsis might be easier to just do this all experiment.
Yeah, well, maize is, of course, one of the most abundantly grown crops in the world, and it's essential for feeding the world. And also because this issue of phenotyping is more of an issue in crops that are grown in large quantities, like maize. Being able to measure a trait of Arabidopsis is relatively easy when you have a small amount in a lab, but maize is grown with hundreds of thousands of plants in one field. So being able to develop a method that can predict the phenotype of each one quickly and efficiently is necessary.
Very cool. Nice job.
Yeah.